Time moves slowly for bones, but the secrets hidden in those bones, especially bones buried in red ochre and sand for tens of millennia, can reshape how we tell the story of humankind. In the outback of Australia, on the windswept shores of a dried-up lake, lie the remains of Mungo Man, a skeleton discovered in the 1970s that has long occupied an almost mythical spot in human prehistory. In 2001, scientists claimed they had unlocked his mitochondrial DNA and that his line diverged from every modern human lineage. Crowds called it a lost human lineage, a challenge to the idea that all people today descended from one migration out of Africa. But then, a decade later, new DNA methods called those claims into question. When researchers revisited the same bone fragments, they found no ancient DNA belonging to Mungo Man. Instead, they uncovered DNA from several modern European sources, evidence of contamination. Meanwhile, a neighboring skeleton revealed an Aboriginal mitochondrial genome, tying it into the living lineage of Australia's first peoples. The result, a scientific reversal, a restored consensus, and a more cautious view of what our bones can really tell us. In this episode, we unravel how an apparent breakthrough became a cautionary tale in ancient DNA, and why Mungo Man's story still matters. The story of Mungo Man begins in one of the most desolate yet historically rich landscapes on Earth. The ancient bed of Lake Mungo lies within the Willandra Lakes region of western New South Wales. Today, it is a dry and windswept expanse, a place of stark dunes and eroded clay ridges. But tens of thousands of years ago, it was a freshwater lake teeming with life, supporting early human communities who hunted, fished, and gathered along its shores. It was here in 1974 that geologist Jim Bowler made a discovery that would forever change the understanding of human antiquity in Australia. While surveying the dunes, he noticed the weathered bones of a skeleton emerging from the shifting sands. Excavation revealed the remains of an adult male, carefully laid out in a shallow grave. Most striking of all, traces of red ochre surrounded the bones, a clear sign that the burial had been deliberate and ritualistic. Radiometric dating later placed the age of the skeleton at around 42,000 years old, making it one of the world's earliest known examples of ceremonial burial. The remains were soon named Lake Mungo III, though the public came to know him as Mungo Man. Unlike a fossilized skull or scattered bone fragments, this was a largely complete skeleton, providing an unusually detailed view of a person from the deep past. His physical characteristics, slender build, elongated limbs, fueled early debates about whether Australia's first inhabitants had distinct regional features or whether they reflected broader patterns seen across early modern humans. For Aboriginal Australians, however, Mungo Man was never just a specimen. He was an ancestor, a man who lived and died on country, and whose burial reflected a continuity of culture that has endured for tens of thousands of years. The discovery resonated deeply with local communities, affirming their unbroken connection to the land. For scientists, Mungo Man represented something equally profound, tangible evidence that humans had been in Australia far earlier than many had dared to imagine. His burial pushed the timeline of human presence on the continent back beyond 40,000 years, aligning with the oldest archaeological traces of Aboriginal occupation. The presence of ochre suggested symbolic behavior, perhaps spiritual beliefs, perhaps social rituals, placing the people of Lake Mungo among the earliest to leave behind evidence of a cultural life, as well as a biological one. Yet, with every answer came new questions. Where had Mungo Man's people come from? Did they descend from a single wave of modern humans leaving Africa, or from multiple migrations? How did they relate to the broader story of human origins? These questions would eventually draw scientists back to his bones decades later, when the tools of genetic science promised to reveal secrets that the skeleton alone could not. It was this pursuit, an attempt to read the deepest layers of ancestry from fragile, ancient DNA, that would transform Mungo Man from an archaeological milestone into the center of one of the most contentious debates in human evolutionary history. By the turn of the 21st century, ancient DNA research was still in its infancy. The technology was rudimentary compared to today's standards, but the promise it held was enormous. If scientists could extract genetic material from bones tens of thousands of years old, they might reconstruct the movements, relationships, and even the identities of ancient peoples. For many, Mungo Man seemed like the perfect candidate for this new frontier. In 2001, a team led by Australian National University researcher Adnan Rizvi and colleagues announced they had successfully recovered mitochondrial DNA, genetic material passed down through the maternal line, from fragments of Mungo Man's bones. According to their analysis, Mungo Man's mitochondrial sequence did not match that of any living human group, nor did it fit into the established branches of the modern human family tree. Instead, the sequence appeared to represent an entirely separate and now extinct lineage of Homo sapiens. If true, the implications were enormous. The findings seemed to challenge the widely accepted out-of-Africa model, which argued that all modern humans today descended from a single population that left Africa around 60 to 70,000 years ago. Mungo Man's DNA, as presented, suggested that when humans arrived in Australia, they may have included groups who represented a more ancient divergence, possibly even predating the main dispersal that gave rise to the rest of humanity. The media quickly seized on the story. Headlines described Mungo Man as belonging to a lost tribe of humans, and some suggested he was proof that modern Aboriginal Australians did not descend from the same lineage as other peoples of the world. The claim stirred controversy, not only in academic circles, but also among Aboriginal communities, who saw the interpretation as undermining their continuous connection to the land. Within the scientific community, the reaction was mixed. Some researchers were intrigued, seeing the results as a bold challenge to established thinking. Others were deeply skeptical. The DNA sequences were incomplete, and the techniques used, based on polymerase chain reaction, PCR, were notoriously vulnerable to contamination. At the time, strict protocols for ancient DNA work had not yet been fully developed, and laboratories lacked the ultra-clean facilities that are now standard. To critics, the unique DNA sequence might not represent a lost lineage at all, but rather a mixture of degraded fragments and modern contamination. Still, the idea of an extinct human lineage in Australia lingered. For years, the 2001 paper was cited as evidence that the story of human origins was more complicated than a simple migration out of Africa. Some even speculated that Australia had been settled by multiple waves of humans, each leaving a genetic imprint that might still be detectable in ancient remains. It would take more than a decade and major advances in DNA sequencing technology before scientists would revisit Mungo Man's bones and put those claims to the test. It also meant that when genuine sequences were recovered, scientists could test them against known patterns of degradation to confirm their authenticity. The field as a whole had matured. By the mid-2000s, complete genomes had been reconstructed for Neanderthals and Denisovans, once thought impossible. These successes raised the bar. Any new claims had to meet the same standards of replication, authentication, and transparency. Against this backdrop, the original 2001 results from Mungo Man looked increasingly fragile. They had not been replicated. The sequences lacked damage patterns typical of ancient DNA, and the methods used at the time would never meet the stricter criteria now demanded by the field. For many, it seemed inevitable that when Mungo Man was retested, the results would either overturn or confirm the earlier claim once and for all. By 2016, the moment arrived. A team of researchers, equipped with the latest methods, returned to Lake Mungo's most famous skeleton to re-examine his genetic story. What they found would not only resolve the debate, but also provide a striking lesson about the pitfalls of early ancient DNA studies. The re-examination of Mungo Man and the successful sequencing of DNA from the nearby WLH4 skeleton had broad implications, not just for the specific debate about an extinct lineage, but for the story of human origins in Australia as a whole. First, it reaffirmed a central point. Aboriginal Australians represent the oldest continuous population outside Africa. Archaeological evidence already placed human settlement of the continent at least 50,000 years ago, with some sites suggesting even earlier dates. The genetic continuity between WLH4 and modern Aboriginal lineages confirmed that those first settlers were the ancestors of today's indigenous peoples. There was no mysterious replacement, no vanished human branch unique to Australia. Instead, the story was one of remarkable persistence and survival. Second, the findings reinforced the out-of-Africa model rather than undermining it. For years, the 2001 claim had been used as an argument for alternative models of human evolution, such as multi-regionalism, the idea that modern humans evolved simultaneously in different parts of the world from earlier populations of Homo erectus. If Mungo Man had truly belonged to an entirely separate lineage, it would have lent weight to that theory. But the refutation of the claim, combined with the WLH4 genome, showed that early Australians fit squarely within the broader framework of modern human expansion out of Africa. They were part of the same migratory wave that gave rise to populations across Asia, Europe, and beyond. Third, the confirmation of Aboriginal continuity carried profound cultural significance. For Indigenous Australians, the scientific evidence aligned with long-held oral traditions, describing an unbroken connection to country dating back to the dreaming. It provided a genetic echo of what communities had always asserted, that they were the first people of Australia, and their roots extended back tens of thousands of years. The study thus served not only as a correction of earlier errors, but also as a recognition of the depth of Aboriginal heritage. Finally, the episode highlighted the difficulty of ancient DNA work in challenging environments like Australia. The fact that WLH4 yielded a usable genome while Mungo Man did not underscore how preservation can vary even between individuals buried in similar conditions. For researchers, it was a reminder that patience and persistence are essential, and that every successful recovery is both rare and invaluable. In the end, the implications were clear. The dream of finding a lost human lineage in Australia dissolved, replaced instead with something more enduring. Evidence that the first Australians were not an anomaly in the human story, but a vital and continuous part of it. Mungo Man, even without DNA, remained a symbol of that deep history, an ancestor who testified to the extraordinary antiquity of human culture and life on the continent. The story of Mungo Man's DNA is more than a tale about one skeleton. It is a case study in how science works, how it sometimes stumbles, and how it eventually self-corrects. The reversal of the 2001 findings carried several important lessons, both for the field of ancient DNA and for the broader public understanding of science. The first lesson is about contamination. Ancient DNA research is inherently vulnerable to modern interference. A single human hair, a skin cell, or even a speck of dust introduced during excavation can overwhelm the faint traces of genetic material left in ancient bones. In the early days of the field, researchers did not fully grasp how pervasive this problem could be. The sequences reported from Mungo Man turned out not to be ancient at all, but rather modern DNA that crept in somewhere along the line. Today, rigorous clean room protocols, authentication criteria, and damage pattern analysis are standard practice. Lessons learned, in part, from cases like this. The second lesson is about the pace of technology. What looked like groundbreaking evidence in 2001 was, within 15 years, revealed to be an artifact of limited methods. PCR-based approaches, once celebrated, could not withstand the scrutiny of high-throughput sequencing and strict authentication. Science is not static. It evolves with its tools. And sometimes, new tools don't just add detail. They overturn old conclusions entirely. The third lesson is about interpretation. Even if the original DNA sequence had been real, jumping to conclusions about an extinct human lineage was a leap beyond the available evidence. Human evolution is complex, but it is also interconnected. Claims that place one population entirely outside the human family tree demand extraordinary proof. In this case, that proof was absent, yet the narrative captured headlines and spread widely. The cautionary point is clear. Extraordinary claims require not only careful science, but also careful communication. Finally, there is a broader lesson about humility. Ancient DNA is a powerful tool, but it is not infallible. Archaeology, anthropology, and oral history remain equally essential in reconstructing the human past. When viewed in isolation, DNA can mislead. When integrated with other lines of evidence, it becomes part of a richer, more reliable story. The Mungo Man controversy reminds us that science is a process, not a set of fixed truths. It makes mistakes, but it also corrects them. And in that cycle of challenge and revision, knowledge deepens. What was once seen as evidence for a lost lineage is now a reminder of the importance of rigor, skepticism, and respect for both the science and the people whose ancestors it studies. The controversy surrounding Mungo Man did not occur in isolation. It unfolded at a time when ancient DNA research was rapidly reshaping our understanding of human prehistory, producing both astonishing discoveries and painful reminders of how fragile the evidence can be. One of the most famous examples came from Siberia in 2010, when researchers sequenced DNA from a tiny finger bone found in Denisova Cave. The result revealed an entirely new group of archaic humans, Denisovans, who had never been identified from skeletal features alone. Their discovery was solid, replicated, and confirmed by nuclear as well as mitochondrial DNA. It stood in stark contrast to the uncertain claims about Mungo Man, showing how ancient DNA could be transformative when applied carefully. Other cases, however, bore closer resemblance to the Australian story. Early attempts to extract DNA from Egyptian mummies, for instance, often produced sequences that turned out to be modern contamination. Claims of very ancient DNA from amber-encased insects, once reported as being tens of millions of years old, were later discredited for the same reason. These episodes highlighted the risks of over-reliance on fragmentary results and the dangers of publishing extraordinary claims without robust verification. The lesson is that ancient DNA, while powerful, is not a silver bullet. It must be read in context alongside archaeology, anthropology, and even oral tradition. Archaeological layers, tool assemblages, burial practices, and skeletal morphology all provide vital information that DNA alone cannot. In Australia, for instance, the presence of ochre burials and long-term site occupation has always been a strong line of evidence for the deep antiquity of Aboriginal cultures. DNA adds another layer, but it does not stand above the rest. At the same time, the successes of the field have been remarkable. Ancient genomes have revealed patterns of migration across the globe, from the peopling of the Americas to the spread of farming in Europe and Asia. They have shown that modern humans interbred with Neanderthals and Denisovans, leaving genetic traces still present in people today. In each case, the power of ancient DNA lay not in single anomalous sequences, but in broad datasets that could be replicated and cross-checked. Mungo Man's story, therefore, fits into a wider narrative. It is a reminder that not every attempt will yield clear answers, that some remains are simply too degraded, and that claims based on weak evidence can mislead the field. But it is also a testament to how much the discipline has advanced, moving from tentative, error-prone beginnings to a mature science capable of rewriting major chapters of human history. Seen in this light, the reversal of Mungo Man's DNA is less a setback than a step in the ongoing process of refinement. It shows both the pitfalls and the promise of ancient DNA, and it underscores why careful standards are crucial when the stakes are nothing less than humanity's shared origin story. The saga of Mungo Man's DNA is a story of promise, controversy, and ultimately correction. What began as a claim of a lost human lineage ended as a reminder of the limits of early genetic science and the importance of rigorous standards. Yet the questions raised by his remains remain far from settled. For one, the absence of authentic DNA from Mungo Man himself leaves gaps that may never be filled. Could future techniques, even more sensitive than today's, one day recover fragments from his bones? Or has time and the harsh environment erased his genetic record forever? The possibility remains open, though each year that passes makes recovery less likely. Another ongoing question concerns the broader genetic history of Australia's first peoples. The WLH4 genome tied ancient remains directly to living Aboriginal Australians, confirming deep continuity. But how much diversity existed among those early populations? Did multiple waves of migration contribute to Australia's settlement, or did a single founding population spread across the continent? Archaeology suggests complex patterns of movement and adaptation, yet the genetic picture is still incomplete. There are also cultural questions that go beyond the laboratory. How should science engage with indigenous communities when studying ancestral remains? The Mungo Man controversy sparked important conversations about respect, consultation, and the role of traditional knowledge alongside scientific research. For many Aboriginal people, the story is not about DNA sequences or models of migration, but about the enduring presence of their ancestors on country. Reconciling these perspectives remains an ongoing challenge for researchers. Finally, the episode is a reminder of science's self-correcting nature. Mistakes were made, conclusions were overstated, but over time, better methods brought clarity. Far from discrediting the field, the correction of the 2001 claim strengthened it, showing that even in the face of error, knowledge can move forward. Mungo Man himself may never yield a genome, but his importance does not rest on genetic data alone. He remains one of the oldest ceremonial burials known anywhere in the world, a testament to the symbolic and cultural lives of early Australians. His discovery continues to inspire debate, reflection, and a deepened respect for the antiquity of humanity on this continent. In the end, Mungo Man's story is not just about what science can tell us. It is also about the humility required to recognize what science cannot yet recover and the responsibility to honor the people and cultures whose pasts we study.